Welcome to the Build Exito and URISE Research Symposium. I'm Tom Keller. I'm the principal investigator for both Build Exito and the URISE program, and I'm delighted to welcome you to this event. The Research Symposium is an important milestone for our scholars. You get to share the impressive research that you've been doing over the past year. You gain skills in reporting and sharing your research, and it's great preparation for future conference presentations and maybe publications. So we have several groups with us today. On the Exito side, we have cohort 6 scholars who are just completing summer immersion. So they have been in their RLC placements for a year, and they will be reporting their research from the past year. We also have cohort 7 scholars who are just finishing up their summer induction, which means they're just beginning their RLC placements, and they get a preview of what it will be like next year when they're presenting at the symposium. And finally, we hope that we have many prospective cohort 8 scholars who may have attended the Summer Research Academy and are in the process of applying for Build Exito right now, so they can see more about what the program is all about and the kind of research that students do. So welcome to all of you. On the URISE side, we have cohort 1, our very first URISE scholars who have been in their research placements for the past year, and they're just coming off of an intensive summer research experience. So they will be sharing their findings with us. And we also have cohort 2 scholars in URISE who have just started their research placements this summer. So they've been in their RLCs for just a couple of months, but we asked them to do their presentations as well because we think it's good experience, and they will be sharing with us the research that they intend to do over the coming year. So another group I hope we have with us today. It would be wonderful to see research mentors who have supported and guided our scholars, and we really appreciate the opportunities that they're providing for our scholars, and we hope that you enjoy seeing and supporting your scholars during the symposium. And I want to also acknowledge some scholars that will be concluding the program soon or already have. First of all, Miriam Ibrahim completed with us in August, and Raisha Paris is completing Exito at the end of immersion, and Emily Mize is completing at the end of fall term. So congratulations to all of those scholars. I also want to thank people that are involved in making this all work, putting on this great event. So first of all, Dr. Decatur Foster has done an excellent job with summer immersion this year, supporting the scholars in preparing their presentations and posters. I also want to thank Dr. Shandy Dixon, who supported immersion, but also is responsible for induction this year. Of course, there are many other people involved in organizing an event like this, so I want to especially note Cassandra Ramirez and Sarah Taylor for helping to coordinate everything, and many others who have helped to pull this all together. So we're very excited to be together in person again. And I want to say, finally, we are very excited about our event on Thursday. So again, the symposium will feature research presentations today, Tuesday, and then the first 2 hours on Thursday. But the last hour on Thursday, just before lunch, we're going to have a poster session and a reception with refreshments. And this year we've combined it with our annual kickoff event to start the new year. So we hope you can all be with us to celebrate. And we've invited many guests from around campus so that they can learn more about Build Exito and URISE programs and see the wonderful research that our scholars are doing. So again, we hope to see you all for that. And We hope you're having a great experience in the program, and we're so proud of all that you've accomplished, and we hope you enjoy the symposium. Thank you.
Good morning, everybody. I am super excited to share just a little bit about what the amazing things my lab is doing up at OHSU in the CDRC with Dr. Emily Quinn, which is also known as the Child Development and Rehabilitation Center. And I'm just going to take a little bit of time today to talk about our new project that we are unfolding now, which is effectiveness of AAC devices on preschool language impairments. So the big question is, what is AAC? Augmentative and Alternative Communication is better known as AAC. AAC in the specifics of our lab serves children and their families between the ages of 24 and 36 months, so little itty bitty babies, that are living with significant language impairments. So this can be things like Down syndrome, as well as autism and autism spectrum disorder. And I wanted to use this slide as just a reference because I'll be using these terms interchangeably throughout the rest of the presentation. So specifically in the context of our lab, this new study is going to be working to address systemic racism in Down syndrome research. For previous context, AAC access has unknowingly been denied to communities of color simply because some AAC studies require families of color to, maybe there's a fee to get involved in this research, or there is lack of information being given out to these families in our communities, or there's just a lack thereof of AAC research going on in general. What we hope to address specifically in our lab, as far as AAC interventions are going on, is to complete this work of creating culturally responsive techniques to advise more children and more families of color to get involved in AAC research. What this will kind of look like is community advisory councils, which will consist of speech language pathologists or SLPs, and families of children who are already using AAC implementation. And we're going to survey them and kind of talk to them about, how can we improve the AAC implementation experience? How can we make it better? What can improve? How can we make it easier for everyone to have this access? We also hope to increase AAC resources and trainings. We also hope to increase the distribution of open enrollments in AAC studies and just make safe spaces for families to be able to come and to help their children along with their language and development acquisition and production. So this is kind of what our study is hoping to look like. As I stated in the previous slide, we hope to start by surveying previous AAC families, and these two next steps will work interchangeably. So as we are surveying these families, we will also be recruiting them for the community advisory councils. As that is going on, then we will use what they're telling us they feel would improve the process to complete the work of changing our process and making it more culturally responsive. Then we will move on to recruitment for our new CAC and ACC study. Then we will move on to implementation studies within the families. So this will look like teaching the families how to use the iPads and tablets within their everyday lives, because it's kind of awkward, to have this device kind of walking around and trying to implement it with your child's daily routine. So we want to make sure that parents have the correct tools to make sure that this is going on positively. Then we will move on to data collection and analyzing and producing our results. And kind of what we're hoping to see for our results is the graph to the right or the left, my right. We hope that over time we see an increase in a lesser amount of sounds that are being uttered by the child and more amounts of words and phrases being invited. So less of things like pointing and more things like go, mom, bye, things like that. But the main point of our project and why I am so excited to be a part of it is just to get an increase of communities of color in AAC and to kind of diversify that field of Down syndrome research. We hope that we can create these safe spaces for families to feel comfortable accessing these resources and also feel confident in using them as well, because that is an important part of diversifying the field. I would like to thank Dr. Emily Quinn and the OHSU CDRC, my senior research assistant, Alex Cook, my graduate research assistant, Kim Curran, just for allowing me to feel safe in that environment and creating a space that is open for me to be able to do this work and be a part of something so powerful and so moving. I would also like to thank the Build Exito team for creating this space where there's diversity and we are diversifying the field of medicine and research, because it's definitely something I've been passionate about doing since I was a little girl. Bureau. And I would also like to thank the NIH for giving us the resources that we need to be able to complete this work. So thank you. Are there any questions?
Yes, it sounds like you're at the beginning stages of some of this. Do you know what's next for the team?
What's next for us is to, right now we're in the process of getting those community advisory councils together. So we're very, very much in the beginning stages. And then once we craft those community advisory councils, we'll be interviewing them via Zoom and kind of getting their take on, you know, how can we improve the system and how can we make it a little easier for parents to get involved.
Will you be able to be a part of that?
Yes. And I've already had a few of those opportunities, and it's absolutely been eye-opening and amazing to talk to these families and also connect with them on a deeper level than just, you know, medical based. It's, you know, it's definitely a relationship based program and study. So I've just loved every part of being a part of it. Question? Okay, thank you.
And our next presenter is Bradley Marks Miller with Dr. Javier Aves at OHSU. They're a Build Cohort 6 scholar, and they'll be presenting the role of medial, PFC, VIP, interneurons, ABC. Just kidding, during reward processing. So I'll let Bradley take it away.
So yeah, sorry, it's kind of a long title, but I'll try to break it down, make it a little bit easier to understand. So the role of PFC, VIP, interneurons during reward processing. The PFC is just kind of where, the location. VIP interneurons are kind of the like mediator, and the what's happening is the reward processing. So the PFC is the prefrontal cortex, sits right in the front of the brain on humans. It's categorizes and selects actions. There's a lot of firing and activity that happens around actions when we're choosing between like a large and a small reward, or you know, just any sort of action. It is a structure that's kind of more developed in mammalian evolution. So we have to use mammal models to study it. We can use like fruit flies, that sort of thing. And then that's on the right-hand side is like the mouse brain, which is the animals that we study. VIP interneurons, they show increased activity around reward and punishment. They're found in the PFC, and they are kind of well situated to help choose between things because they synapse onto both excitatory and inhibitory neurons. So basically, like they can turn one thing on and one thing off at the same time by receiving like a single input. To test that, we use a behavior that we've come up with where we train them to flexibly choose between rewards, a large and a small part. In this case, there's like a large reward on the right side, a small reward on the left side. They figure out that the large reward's on the right side. They go there pretty quickly. They learn how to do that. After 7 visits or some other variable, we switch the sides, and it may go to the right port where it knew the reward was higher before, you know, 2 or 3 times, but then it usually quickly figures out and switches to the left. Oh no. Well, there is a video that I will work on maybe approving on my phone to show at the end during questions, but we'll move on from there. So again, the hypothesis is that VIP and interneurons and the PFC help mediate that reward behavior. We teach them how to do the behavior, and then we inject them with a virus that expresses a protein in those neurons, which allows us to shine a laser on them, and it turns off those neurons. So we can do the behavior both with and without the VIPs being active. This is an example of one of the probes I made. You can see the fiber that sticks out, that's where we attach the laser. And then the little wires that come down, those are the fibers or the wires that pick up the electrical signals. That's like an example of the readout that we get on the computer. You know, yeah. So just kind of brief results. We have more, but just more digestible results. On the left-hand side there, the y-axis is the average number of trials to switch. So that's like after it's, you know, after the reward has switched sides, how many times it goes to the wrong port. And normally it's around 2.5. When we turn the laser on, that's inhibiting the VIPs, so kind of like removing them from the equation. It increases about 1 poke on average. And then just to kind of reiterate that the mice learn the task pretty quickly, about 2 weeks training. And then when we inhibit the VIPs, they switch slower. They don't incorporate the reward information as readily. And then I just wanted to quickly thank Dr. Thera Boss. He's my PI. Dr. Alex Sonnenborn, he's the postdoc that I work probably most closely with. And then Randall and Lowell are other guys in the lab. They don't really do much on this project, but they do help morally, I guess. Okay. Are there any questions? I'm gonna try to prove that so you can see the video. Yeah.
So for the oxygen lights, are you shining it on the nerve?
Yeah. So so I can go back here. Yeah, so we like do a craniectomy. So like we drill a hole in the mouse's brain and then lower this this into the brain, into the PFC. And so it's a laser on both sides of the brain. That's why there's like a dual fibers there. And yeah, we shine laser. It kind of like how you can shine a light through your skin. It goes through the brain pretty far. Mouse brains are real small, so yeah, you can get pretty good coverage.
In my lab, we also do oxygen lights and we shine on the vagus nerve. So I was just wondering what nerve, you know, what nerve. Or is it just like the PFC and all the nerves?
Yeah, so it's just like the PFC and all the nerves, and then so yeah, it's like just the general like inside of the PFC, yeah, and then it's the virus that we saw. It's like a AAV virus, and it puts channel archirhodopsin. Here I can show. Sorry. Yeah, it puts in like archirhodopsin things in the membrane, and then when the light shines on, it kind of pumps out hydrogen ion, so it keeps it from firing. Any other questions?
Naive question. So all of the teeny little data pieces, or what's going on in the left graphic?
Oh yeah, so that's the actual virus that we like when we order the virus. That's how we order it. And so like this is like GFP. That's like the green fluorescent protein. That's pretty common in a lot of biology things. And then this is actually the arch, the units for arch. And basically, like it uses this lock site to there's the mice we use are they have a pre-site and genetically modified into VIP cells. So that's what that targets. So the virus goes in, it finds that Cree, switches the locks and puts the genes that are in there into those particular types of cells and not other cells.
Okay.
So it kind of like uses that to.
It's like the combination.
Yeah. And then this, it might not work. Oh, it did. So here is an example of the mice actually performing the task. That's them going to the right side. And then after it goes through, I think this one does like 8 times, and then it'll switch to the left side and continue going.
What's the reward?
So it's water. They're water restricted. Yeah. So we take them to like 80 to 75% body weight of water, and then they just get water.
I think there's a question.
Oh, I'm so sorry.
So how is this?
Yeah, so I got interested in it largely because VIPs, there's a genetic variation associated with schizophrenia. But there's a lot of things about like decision making, obviously like alcohol or other addiction issues, just impulse control issues, ADHD, a lot of things have to do with how we make decisions, obviously. So, but that's the primary one that I'm interested in is schizophrenia.
That'd be cool. But I mean, this really does, I mean, this is just giving some understanding how that works and that work.
Yeah, trying to figure out the mechanisms that we incorporate those rewards back in to the decision process.
Is there different ways that you could reward and you, for instance, be able to try to see a place?
Yeah, absolutely. That's what we're currently doing. I'm like doing surgeries this week for mice that we're training on doing like a large, and this is a large and no reward. We're doing like a large and a small reward. And then towards the end, we're gonna like increase that and see how long, or like, you know, make the difference smaller and see how small of a difference we can have before, you know, we see an effect, and does the laser on or off, you know, affect that.
I have one question.
Yeah.
How many mice were used for this?
So the first cohort, we used 8, generally somewhere between 8 and 10. I try to get a mix of females and males, but yeah, it depends. The surgeries, the females don't always handle it as well. I think the ISO, like the stuff we use, sometimes is a little harsh. So, or maybe I've been getting better at turning it down a lot more. It's like you don't want them to feel pain, but you also, you know, want them to make it through the surgery.
Yeah.
So, yeah, anything else? Oh, thanks.
We've got Emily Mize, and their PI is Dr. Matt Drake at OHSU. They're a Build C6 scholar presenting, I might butcher this, the impact of eosinophils on purine receptor, in mouse sensory neurons. So with that amazing introduction, I'll let Emily introduce themselves.
Hi everyone, my name is Emily Mize. I work in Matthew Drake's lab. We study the neurobiology and neurophysiology of asthma, and I'm talking to you a little bit about the research I've been doing for last year on the effect of eosinophils on P2X3 purine receptor expression in mouse sensory neurons. So asthma affects over 300 million people worldwide. It's characterized by excessive bronchoconstriction, airway hyperreactivity, and eosinophilic inflammation. As you can see on the left, these are blown up airways, and you can see on the left a healthy airway, which has a lot of space for the air to flow through, whereas an asthmatic airway has a lot of inflammation caused by white blood cells, especially these eosinophils. So airway nerves control bronchoconstriction. And to orient you to this figure, up here we have the brain stem, the parasympathetic nerves, which synapse on airway smooth muscle, and the sensory nerves, which synapse on this airway epithelium, send messages up the vagus nerve into vagal ganglia and back to the brainstem. So P2X3 receptors synapse here on these sensory nerves in the airway epithelium. They bind to ATP and induce cough and bronchoconstriction. So P2X3 really triggers a sensory nerve activation, and it's highly involved in pain, cough, and bronchoconstriction. So eosinophil-induced airway nerve dysfunction worsens bronchoconstriction in asthma. As you can see in these images taken before I got into the lab, the eosinophils are stained in red and the nerve fibers are stained in black. And you can see these eosinophils really integrate themselves into the nerve structure, which worsens this bronchoconstriction in asthma. So my research question I've been working on is, do eosinophils increase P2X3 purine receptor expression in these sensory neurons? To study this, I have 4 mouse models: wild type mice, which have a normal level of eosinophils, Phil mice, which have no eosinophils at all, NJ17 mice, which have airway eosinophilia, and NJ16 mice, which have systemic eosinophilia. To test this, I used RNA-scope, which is an in situ hybridization method, which labels P2X3 mRNA, and I did this in sensory nerve ganglia. So these Z probes bind specifically to P2X3 and then undergo serial amplification here. And then the mRNA of interest is identified in red, as shown here. So this is a vagal ganglia, a sensory nerve ganglia. The red is P2X3 mRNA, and as you can see, it's very highly expressed in these sensory nerve ganglia. After analyzing this data, I found that P2X3 expression is increased in airway sensory nerves of eosinophilic mice. On the y-axis here, we have the percent area positive for P2X3 staining. On the x-axis, again, we have these mouse models. So we have wild type mice, fill mice with no eosinophils, NJ16 mice with systemic eosinophilia, and NJ17 mice with airway eosinophilia. As you can see, there's about a 20% increase in the medians between wild type and NJ17 mice, which leads us to believe that eosinophils may positively regulate P2X3 expression. So eosinophil-induced changes in P2X3 expression may underlie increased ATP sensitivity in asthma. So humans with asthma that inhale ATP actually have higher response to it and cough more than humans without asthma. And so this may be due to the increase in P2X3 expression in humans with asthma. Also, P2X3 may offer a new therapeutic target for bronchoconstriction in cough and asthma. And there's currently a P2X3 antagonist in clinical development for chronic cough. And I'm trying to connect P2X3 to asthma as well, so that that can be another treatment for humans with asthma. Currently, I'm working on analyzing P2X3 protein expression using antibody staining. So I've been doing mRNA so far. I'm working on protein now. I'm also going to test the significance of differences in expression on the airway physiology of mice in vivo. And I'll also extend my observations into a model of allergen-induced airway eosinophilia rather than chronic eosinophilia. And thank you to all the members of my lab and Build Exito and the Ford Family Foundation and the NIH and everyone who came here today. Any questions? Yes.
Hi, thank you. Great presentation.
Thanks.
Are you testing both female and male mice?
Yes.
And if so, do you see any sex differences?
So I am testing an equal amount of female and male mice. And the differences that I've seen so far haven't been in P2X3 expression, but I've actually found that there's a difference in mouse weight. And so males usually weigh more than females, but the males that have eosinophils actually weigh less than the males that don't have eosinophilia, but females, there's no difference. So I'm not sure why that is, and if I have the time to study it, I might, but it is kind of interesting that there's a difference in males and not females.
Thank you.
Thank you for the question. Yes.
So is it just I guess breathing in ATP that is the turns off the irrigation, or is it like in the body production?
Yeah, that's a really good question. So yeah, it's usually in the body. The reason that we have humans inhale ATP or mice inhale ATP is just to try to replicate that, but usually in the body. So what happens is, cells store a lot of ATP in them, and when they undergo cell stress, they lyse and release that ATP, and that's what causes the cough and bronchoconstriction internally. And so, yeah, mostly the ATP is coming from within the lungs. That's a good question. Thank you.
Our next presenter is Jenny Tran. They're part of Dr. Ken Stedman's lab at PSU. They're also part of the Build Cohort 6 scholars. And the presentation is mutant viruses from hell, comparing the capsid structure of BP1 mutants to the wild type. So I'll let Jenny take it away.
So hi, my name is Jenny Tran, and I will be presenting mutant viruses from hell, comparing the capsid structure of BP1 mutants to the wild type. So viruses in general can be described as genetic material tightly packed into a protein coat called the capsid protein. And in this case, we are focusing on SSVs, so Saccharomyces spindle-shaped viruses, and we're focusing on Saccharomyces spindle-shaped virus 1. These viruses live in extreme conditions at a pH of 3 and a hot temperature of 80 degrees Celsius. They're also found all over the world in hot springs, from Japan, Russia to the US. And you can see on the left image there is a 3D model of SSV1 in front of a hot spring in Japan where SSV1 was first discovered. And on the right over here is a hot spring in Lassen volcanic national park where we recently took samples in the hopes of finding the host of the SSV1, which is Saccharomyces leucotericus, a type of archaea. It is still unknown as to how SSVs can withstand these extreme conditions, especially because they have the same amino acids as us. Which is why we hope that with further research, we can begin to answer this question and also learn more about viral evolution, as well as early life on Earth, since earlier conditions were believed to be the same as where these SSVs are currently found. So there are two main capsid proteins that make up the capsid structure of SSV1, which are VP1 and VP3. However, the VP1 is known to be essential for the virus to be functional, while VP3 is not. And in VP1, at position 66, you can see, sorry, in the image, you can see that the black arrow is a glutamate at position 66, and it's a VP1 sequence all aligned with other SSVs. You can see that that glutamate is highly conserved in all the SSVs. And at the same position, there's a proteolytic cleavage site where the protein is cut so that the virus becomes functional, which brings to the question of how the glutamate is important towards this virus. And to determine this importance, we have substituted this glutamate at this position with other amino acids. And so because many of these mutants have already been made, we were able to grow them out from freezer stocks in 50 milliliters of yeast sucrose media and put them in a shaking incubator to grow at 80 degrees Celsius for about 2 to 4 days. We then separated the cell pellet from the supernatant, which is the liquid above where the virions will be, and spun them down in a centrifuge for 15 to 20 minutes, and then concentrated the virions in Vivaspin 6 milliliter concentrator tubes. We then prepped the virions in a copper grid that was used for the transmission electron microscope to image these virions. And once we got these images, we measured the length and width of 35 virions for the wild type as well as the mutant using the ImageJ application, and ran two-tailed t-tests to determine if the mutants were statistically different from the wild type. And you can see here on the graph, it's showing the average length of the virions for the different mutants as well as the wild type, shown by the asterisks above. And you can see that the glutamine, alanine, glycine, and leucine mutants were statistically different in terms of length. The glutamine was statistically different, in which it was longer than the wild type, while the leucine was significantly shorter in length, while the alanine and glycine were statistically different in which they were longer as well, but not as much as the glutamine. In terms of width, only the glutamine mutant had a statistical difference in width in which it was significantly smaller, while the rest of the mutants did not display any statistical differences. And here are just some images taken from the TEM of each mutant that we have so far. You can see that in some of the images, like the glutamine, some of the virions are significantly longer in length and also have a smaller width compared to the virions in the wild type. And you can also see that there are some mutants that have like similar width and length compared to the wild type, like in the methionine mutant here. And in conclusion, the glutamine mutant seemed to be the only mutant that had a statistical difference in both length and width, while the mutants of the alanine, glycine, and leucine were statistically different in length. I would like to thank everyone in my lab, especially Dr. Stedman, Nacho, Jono, Tijani, and Denise, as well as NIH and Bovexito for the provided grants down below. These are the references I used for this presentation. And thank you everyone for listening. Does anyone have any questions?
Do you have a plan to apply this to the line for the purpose of
So in general, it's these viruses, like I was mentioning earlier, live in hot springs and they can withstand like really acidic and really hot temperatures. And it's interesting because they have the same amino acids as us and they can withstand these conditions and we cannot. So we're hoping to study more about that capsid structure, the proteins, VP1 and VP3, to understand pretty much why they can withstand these conditions and why we cannot. And originally, when we made these mutants to begin with, we didn't expect the virus to be functional. So it was really cool that it was, which is why I started the project of just comparing the length and width to see if they had any differences with the wild type.
Do you know, like, does the width or length, does it have any, I guess, stability of the virus? Like, I guess, I don't know, are you gonna test them in like conditions that?
So we are still in the process of making the remaining mutants. And hopefully by then we're going to use what is called halo assays, which is just like a plate of the host cell, Saccharomyces bactericus, and just dropping mutants down to see how effective they are. If that answers your question.
Yeah. No, that's good. Yeah, like just to see like how that affects.
So that's one of the things that we're hoping to do in the future. At least right now, I'm just trying to get the rest of the mutants, imaging them, and then hopefully testing the infectivity.
Yeah, what do you do?
So right now I am finishing up the project of making the mutants to begin with. So the person before me, Tajani, she was a Build Exeter student who started the project of making these VP1 mutants. And so she has most of the DNA already. So what I'm currently trying to do now is take the mutant plasmids and inserting them into the host cell to grow out and then image. Any more questions? Yes.
Jenny, maybe just follow up on that question. So what kind of day-to-day things are you doing in the lab?
So
Applications, you know, sort of, I think that is that sort of following up on your question,
Yeah.
Things that how are we doing? Scar, are we doing? Cover skills, those kind of things.
So right now I'm growing out the host cells and making sure that first of all they're growing because sometimes they don't. But and then I prepped them for basically getting the mutant plasmids into the host cell with a process called electroporation. So essentially we shock the host cell to make the cell wall more permeable to allow that mutant plasmid to be in there. And then we let them grow in a shaken incubator about 2 to 4 days. And then we run it through a supernatant PCR, stands for polymerase chain reaction. So we're replicating that DNA, hopefully that it's there, and seeing that if it shows, and if it does, then we grow them out in 50 milliliters of yeast sucrose and essentially do what I did with the methods earlier of just spinning the virions down and concentrating them to image with the TDF. Does that answer your question a little bit better? Yes.
This is not a silly one, but like, what do y'all do when you're done with your sample? Like how do you?
Where
does it go? You know,
So just to be clear, SSV1 or SSVs in general only infect Saccharomyces cells, so they don't infect humans. So let's put that out there. But we just bleach them pretty much and let them sit for a while, and then can just pour it down the drain and wash flasks and everything. Any more questions?
Center, name is Ashish Ramtal with Dr. Hadashriya Agarwal from OHSU. He's a Bill Sick scholar, and I'll let them pronounce their title because I'm going to butcher it. So take it away.
Hi, everyone. Thanks so much for being here. My name is Ashish, and I'm really happy to have this opportunity to tell you a little bit about the work that I've been able to do for the past year. So without further ado, the title of this great presentation is interleukin-1 receptor 1 gene. Sorry, my friend over there is just laughing at me. So it's interleukin-1 receptor 1 genetic knockout, which rescues aberrant myeloid biasing and TET2 knockout hematopoiesis. It's a big title, but essentially, I think at the end we'll be able to understand it a little bit more. So what are the mechanisms driving TET2 clonal hematopoiesis, and how does blood differentiate in aging mice, and how does interleukin-1 contribute to clonal hematopoiesis? These are the three overarching questions that I really looked at over the year. So clonal hematopoiesis is a risk factor for the development of heart disease, and not just heart disease, we have leukemia and diabetes. And so the more inflammation and the more mutation you have in the specific gene called TET2, which is a DNA demethylation gene, the more likely you're going to suffer from these diseases and not be able to fight the good fight. So people with over the age of 60 has a 10% chance of having clonal hematopoiesis already. And we want to be able to understand what we can do to decrease the amount of clonal hematopoiesis, essentially. So myelodysplastic syndrome is a syndrome where immature blood cells in the bone marrow do not become mature. It's one of the main things that I've been able to see in my research. So again, this is a diagram of inflammation. So as you age, the more inflammation you're going to get in your body, and the more mutations you're going to have with TET2, and the more likely you're going to develop AML or MDS. So in terms of the inflammation that we used, we looked specifically at IL-1, B1, potent inflammatory cytokines. My research really focused on, obviously, TET2 and how that leads to pre-leukemic states in inflammation. So I'm sure you guys have all looked at blood and stuff like that in biology, but essentially monocytes and neutrophils or myeloids, they have the ability to develop into MDS, and lymphocytes are white blood cells also, but these are like B and T lymphocytes, where if you don't have as much of these, it's a lot harder for your immune system to fight lastly, actually, let's continue. So the hematopoiesis hierarchy, this is a very in-depth hierarchy of what I look at. So essentially what happens is you have stem cells that are found in the bone marrow, and they differentiate into different mature blood cells. And depending on what happens with the mutation, you can either have too much T cells or too much B cells. And we want to be able to get all of those cells in normal conditions. So I looked at mice that were over the age of 4 months, and that's like an age study essentially. I looked at wild type interleukin-1 knockout, which is basically the inflammatory signaling, and TET2 knockout, which is the mutation, obviously I talked about, and then double knockout, which doesn't have interleukin-1 or TET2 knockout. So you can see that the white blood cells, the TET2 knockout is significantly lower, and that indicates that it's MDS-like. And the double knockout, so if you take out the signaling as well as the TET2 mutation before the mice becomes older, you can essentially almost have it as normal, right? So lymphocytes, again, decrease exponentially, but the DKO was able to assist in bringing it back to normal. And in terms of red blood cells, it's significant enough that the double knockout is helping. As for monocytes, we see that there's a lot higher expanded rate of monocytes in terms of TET2 knockout and neutrophils. And the double knockout does do a significantly good job of lowering that to a normal state. So just in conclusion, just reiterating what I talked about. The wild blood types, white blood cells reduced by TET2 knockout, and then the myeloid expanded in TET2 knockout. And so what we're going to be able to focus on going forward is looking at potential therapeutics for cardiovascular disease and clonal hematopoiesis in terms of interleukin-1 receptor 1 signaling. So we found out that the double knockout does a really good job of making sure that you can have like a normal cell. And so this is an H study that I was able to conduct very closely with John McClatchy and my PI, Dr. Agarwal. And I've been so blessed to have had the opportunity to work with them. Obviously, this is just a small snippet of what I've been able to do. We look at various other systems like flow cytometry and antibodies and just so much more. But this is just a small glimpse, and I've been able to share with you. And so thank you so much for being a good audience.
After you've knocked out the FG for a mouse, observe that their white blood cell counts are normal. The further step is see if there's a susceptibility to getting other diseases.
So I mean, like, I guess like with wild type, they're just normal cells, but then with TET2, that's a mutation in the DNA. So if you have like more mutation, then you're obviously going to have more chances of other diseases. So essentially, like this is a gene, and so when that gene is like, you know, so TET2 knockout means like there's a mutation and that gene has stopped working. And so when that gene has stopped working, that's going to obviously affect all kinds of other factors. And that's why we like the research that I do is not just on blood cancer, because obviously AML is a cancer, but you have MDS, which is like your blood just stops working and becoming mature. And so it can go to so many other different areas. That answers your question. Yeah. Yeah, so like essentially like there's so much to learn and there's just so much that we don't know in vivo. And so like sometimes I'll be doing experiments that like we like have like no idea about. And so we're just like kind of seeing what's going to work and what's not going to work. And just I think that's what's really exciting, because I'm like, dude, I have no idea what I'm doing. Or like, wait, okay, I do have an idea. But now it's like, wait, we're doing something else. And so it's really good, but yeah.
So what characterizes a pre-leukemic state?
Yeah, so pre-leukemic state is essentially like when you're like the TET2 mutation is like really taken out. So yeah, just like the mutation itself.
Yeah, like symptom wise, you know, like is it just like less white blood cells, more white blood cells?
Yeah, so I think less white blood cells. Yeah. Yeah, because MDS is the pre-leukemic state. And so essentially my data showed that TET2 knockout has less white blood cells, and that indicates that it's pre-leukemic.
Did you work with both male and female mice?
Yeah, so we both worked with both of them, but
Any
like
differences?
no, so we didn't like look specifically on the differences yet, because it's just such a broad topic to look at. So I think well, we did obviously section it out, but like I didn't look at the data analysis of like what's different, what's not yet.
Okay, so the data is there, just haven't analyzed.
Yeah, because like I think the more important at the moment is just understanding what's happening.
Thank you.
Yeah.
All right. Okay, so we'll move on to our next presenter, Helena Tsai, who worked with Dr. Carolyn Kwong at PSU, part of Build's cohort 6, and their presentation is called Factors Impacting heritage language maintenance for children. So I'll take over.
Thank you. Hello, my name is Selena Sai. I'm part of cohort 6, and I'm currently conducting research with PSU's Child Language Learning Center under PI Dr. Carolyn Kwam. This current systematic review article that we're working on is centered around the factors that impact heritage language maintenance in children. So we're going to start by defining heritage language. Heritage language is a minority language learned by speakers at home as children. So for example, if your grandparents spoke Russian at home and then your father spoke Russian, your heritage language would be Russian. And so in the US, there are 381 languages other than English that are spoken. And the recent number of heritage language speakers has been growing at a faster rate than usual, which is faster rate than the growth of the US population, an average of 26.98% per decade compared to 10.88% per decade from 1980 to 2010. So this image reflects a finding we found in previous research, which discovered that there's often a 3-generational linguistic pattern that exists for language shifts. So the first generation is fully fluent and utilizes the native language at home. The second generation is considered transitional. They speak the heritage language at home, but not at school or out, not sort of socially outside of the home. And then the third generation, the native language is no longer spoken either at home or in wider public spaces. So what is the significance of this research? Previous studies have found that parents exhibit fears of passing on their heritage languages due to their own experiences with discrimination, shown in this chart here, the perception of their heritage language as unvalued in American society, accents in broken English, and racial bias and discrimination from their language. It is unknown what the nature of the relationship between a heritage language's language status in society and language maintenance across generations is, and how information received from community members influence parental beliefs about passing their heritage languages to their children. So knowing the relationship between a language's social status and the maintenance of said language can impact the preservation of the endangered languages as well as cultural traditions. So we are conducting a systematic review article. So we are currently investigating literature, We're currently doing our searches right now and going through the data and excluding articles based on our criteria. To the right is Prisma. This is not updated for our current amount of articles. We're running a second search right now. But this is from our previous data set, where you can see that we started off with 5,203 articles and narrowed it down to 38. Right now, I believe that our current search came up with around 1,000 results, and we're narrowing that down as well. So searches were performed in 4 electronic databases: Google Scholar, Web of Science, ProQuest, and EBSCO. For articles related to heritage language maintenance and heritage language loss in relation to parental beliefs, outside influences, and the language status in society. The literature included will be reviewed for patterns related to parent perspectives on language status and benefits of maintaining their heritage language throughout generations. So we found that Spanish and Chinese remain the 2 most common groups of heritage languages in the United States. However, in the United States, English dominates the public school system. Due to the sheer size of the linguistic populations, these 2 languages achieve the closest language status in comparison to English in American society when compared to other heritage languages. Often when parents are prioritizing academics for their children, they often feel pressure to promote that child to English language development as opposed to any other languages or their heritage language. And another one of the themes that emerge across several papers is a strong association between heritage language proficiency and communication and cohesion with the family. While parents strive to support their children's attachment to their heritage, culture, and community, they also seek to ensure their integration into mainstream society via this emphasis on the English language. And families across ethnicities expressed a desire for children to participate and interact with individuals in the broader social community. So here's our references, and our funding was granted by the Build Exeter program. Any questions? Oh.
So is there a reason that Spanish and Chinese are more common as like a heritage language that's maintained? Is it just due to the amount of population or that's more common in the languages around?
I definitely feel like it's a population influence. A lot of the articles that have come up have been centered around, like from what I've been looking through in the articles that have been coming up in this search and in the past search, mostly Korean, Mandarin, and Russian have been coming up, I feel like, as the top three actually, and then Spanish, obviously. So I do think it's more of, that's where the research has been based previously, and also that is like a large amount of the population as well.
Yeah, kind of a bit of a follow-up to that as well, but you see a difference between Spanish and Chinese in terms of sort of value, and part of mention what you mean by that, but
Yeah.
your poster previously, there was something, there's a bit of a difference.
Yeah, I definitely think there's a lot of like social factors that go into that. I think that Spanish and Chinese kind of go through waves of like the way that we as Americans perceive those languages. Like with the pandemic, there was a lot of bias against Asians. And so that influenced whether or not, obviously, whether or not people feel comfortable speaking their heritage language in public. And the same could be said for Spanish in certain contexts. So I definitely think that the impact kind of goes through an ebb and flow depending on how we as a society are viewing that group of people at any given time.
Are you gonna plan to incorporate that into your review article?
I'm not sure. I think it would be an interesting thing to consider because it definitely does have a large impact on, I feel like it is part of that community influence, is that sort of aspect of it. So I definitely think it would be good.
It's a very, current topic.
Very much so, yeah. Cool.
Thank you.
So, because earlier you mentioned that some factors that lead to not being English languages in this country than social stigma, right? Which that might be, is that more important, the social stigma of these languages, is that more impactful than, schools we use English here, but it's mostly the social aspect. Where there's that, and so then it's interesting, just then in your lab, then if you think that about, and it's kind of about capitalism. You can use this for like colonization because, those will sort of tie into those situations. But are those, so with your results, like Shannon, if you have the last, so what are the key words? So what are the factors that impact low maintenance?
Like, so what are these factors?
Yeah, so like what are the words that come up in the results or that come up, even if it's not, even if it's just
Yeah, I feel like from what I've seen in the articles that have been coming up, there's a lot of discussion about like the impact that the school system has on parental decisions. I feel like that's been coming up a lot. And also like, I'm trying to think, sorry.
but then there is a chance the impact of the school. Like,
Mm.
sort of like, I mean, you just see that so many for the parents, because they're worried that their child's not going to do that. So they just focus on it.
Yeah, it's more a worry about them being able to succeed going forward if they don't place an emphasis on English as opposed to their heritage language. So it's like, yes, maintaining their heritage language would be good in a social aspect of their community, but would it be a positive impact on their ability to succeed, like in academic senses, I guess?
It's like the comments, interesting. That like people always said now that it's not one of the languages that people want to hear, language that you wish you wanted the other, it's good. just about the years of observing. It's not really the case throughout the pattern. So it's interesting.
Yes.
What's like the earliest paper? Because I didn't know this type of research existed. Like, I'm a third generation and I follow that third generation where I
Me
don't
too.
speak Tagalog, which is Filipino. Like my parents did, and then, I didn't learn or wasn't taught it growing up. And so I definitely feel that cut from the cultural tie to like the Philippines. So yeah, it's just cool to know that the research exists, but I don't know if there's a novel paper out there that started like recently. It sounds like it's very recent.
We started, the articles that we included started in 2005 or 2005. Yeah. Yes.
We were interested in finding more recent papers. So it doesn't mean that there would be older scholarship. Yeah, I've been a quantitative researcher, like measuring language proficiency, and so this is also new for me. So, so I can tell you about some of the authors of the paper. Betty Yu was a big one for me. She's in San Francisco and she studies Chinese speaking families. Looks at the intersectionality with disability as well.
And their presentation is called Behaviorist Approach to Suicide Prevention. They work with Dr. Antonia Alvarez's lab, and they're also a Build scholar. So I will let Jessica close us out.
Okay, thank you. Hi, everybody. Okay, so yes, behaviorist approach to suicide prevention, more specifically operant conditioning, and my name is Jessica. So for our research, our purpose is to collaborate and create an equitable suicide prevention solutions that work with young people in a virtual environment. This objective has been initiated during a global pandemic and public health crisis, and when within a national movement to address structural and systematic violence, and will continue throughout the many stages of the aftermath of these events. We recognize that young people have unique needs in virtual spaces and commit to co-navigating these spaces through adaptation, creativity, reflection, and a willingness to grow, learn, stretch in our approaches to interventions and best meet the needs of our youth. So some background information, we collaborated with the Oregon Health Authority, the Association of Oregon Community Mental Health Providers, and the Oregon Pediatric Society. And this research was brought on by the global pandemic, and mental health providers were having a difficult time transitioning to a virtual setting. So we wanted to develop curriculum that was appropriate for the youth in Oregon, especially since we had some of the strictest policies for COVID-19. And I'm interested in this qualitative research in examining operant conditioning and suicide prevention. So this is the ABCs, which is the antecedents, events that lead to, the behaviors, actions that result in, and the consequences, either reinforcing or punishing those affecting future behaviors. So some methods and materials, we had 8 young adults, 18 to 24. We invited them to a task force for about 12 weeks, and they provided feedback on the Youth Save curriculum. And this was the part where I was interested in. We had young adults, 25 of them. We invited them to a focus group, and we interviewed them, their personal experiences with suicide assessments, safety planning, and their thoughts about suicide interventions. And they provided explicit feedback and the processes and tools that adults and providers in their community might have used to support them. So with that qualitative research, I kind of examined that, and this is my methods. I did a qualitative analysis. I reviewed the interviews with the subjects from the Youth Save collection. I wrote down key themes and memos from the interviews, and I chose a inductive process. So I identified the antecedents that led to the behaviors and the consequences. And I reviewed recent published peer-reviewed literature on operant conditioning and suicide prevention. And I determined some maybe future directions and implications, possibly methods to form a better suicide prevention curriculum for youth and minorities. And I identified some limitations that could hinder this process, especially like self-reporting. There's probably some stigma with that and not likely to disclose their thoughts about suicide and what they had their personal experiences with that versus field observations. So some future implications, when I looked at a recent research by Nock et al., he had people with PDAs report their triggers, their thoughts, their feelings in real time, which is an ecological method. I found that really super interesting because it's kind of a new technique in clinical psychology. So these antecedents that led to the behaviors have consequences. And so what I found from the qualitative research from the Youth Save curriculum development was that there was a lot of themes that triggered people. And I think that we should consider in the moment solutions for these that cause SITBs, which is self-injurious thoughts and behaviors, instead of relying heavily on self-reporting. So these in the moment solutions should especially be addressed while considering marginalized communities and identities. So special thanks to Dr. Alvarez, and this research is brought to you by Build Exito. Thank you. Any questions?
Can you describe a bit about antecedents and what
Yeah,
that is?
I have a hard time saying that. So that's like triggers, maybe like a breakup with your boyfriend or girlfriend, maybe a transition in your life, like going to college all by yourself, moving away from your family, isolation, stuff like that, job loss. Mm-hmm. Go ahead.
So was the Youth Safe program, was that something that had already been developed, or was that a curriculum that you put together as a part of this?
Okay, so I came in during the summertime. Actually, I transitioned over to this lab, but what I gathered from what I read was that this was already in place, but they were kind of like seeing what other people thought about it as a virtual curriculum. Go ahead.
The sample size that you brought in, that was 25 participants.
Yeah, that was the qualitative part, yeah.
And so the research that you're looking at, were those people who were giving feedback, like have suicidal ideations, or were they just kind of?
Yeah, so I think we did like a snowballing technique where people identified themselves as having that, and then they asked, we asked if they knew anybody else within their circle that might be interested in this. So yeah, they identified as having that. 18 to 24, I believe. Yes. Okay.
Yeah, I was curious about what some of those of the just-in-time interventions were as far as antecedents are concerned.
Antecedents, thank you,
Yeah,
that's a hard
and
word.
I was curious what those would be and how you could implement them. So the curriculum, I see that as being much more about, so it goes back to the kind of thing, it's supposed to immediate reaction. So what are some of the ideas of how that could work?
Man, so that's like a future direction implications for how to go forward. I don't really know, but I think that there should be a focus on marginalized communities instead of mainstream curriculum development. Also, what I read from a study that was done at Harvard, they use PDAs, so maybe possibly tracking through smartphone apps. Is that what you were?
Yeah, it's curious if that's kind of thing they were doing.
Yeah.
Again, I think it's a great idea, but.
I know, it's kind of hard.
Yeah.
I mean, a lot of people, I mean, how do you, do a field observation of like how people think? You really can. We rely so heavily on self-reporting, but that's also really limited. So, go ahead.
Is there any kind of like difference like methods that we have to do for people who have like disabilities that have ideation more frequently? Is there any way you could like those kinds of people? This is more common. They probably track the triggers a lot.
Yeah, I think that's I mean, people did identify what if they had a disability. A lot of people said, I don't have that here. A lot of them expressed anxiety and depression. Some of them had borderline personality disorder. And so you can kind of see DBT, if you ever heard of that. There a lot of this operant conditioning also is in DBT, where you write down what was bothering you that day, what led to this behavior. So I don't know if we should necessarily just focus on people with those disabilities, but I mean, it's not a bad idea. All right. Anybody else? Thank you.